Okay, let's get started. So first of all, thank all attendees from different countries for joining today's seminar. I'm Jiaming Chen from Institute Pasteur of Shanghai, Chinese Academy of Sciences. Today, Rachel and me will moderate the next two seminars. So first, let me introduce today's first speaker, Dr. Youbao Zhao from he recently was recruited as a full professor at the Department of Veterinary, Henan Agricultural University in China. Youbao got his bachelor degree in 2006 from Shenyang University of Technology, master degree in 2009 at Dalian Jiaotong University. He finished his doctoral training in 2014 at the Institute of Microbiology, Chinese Academy of Sciences. From 2014 to 2020, he was in Xiaolonglin's lab at Texas A&M University and the University of Georgia as a postdoc to study mechanisms involved in Cryptococcus neoformans infection. So Youbao published a series of excellent papers in high-profile journals such as the Carbon Biology, MBio, and PLOS Genetics, etc. And today, the title of his talk will be Adaptation of Cryptococcus neoformans to genotoxic stress and hypoxia. So, okay, now Youbao, it's your time, and let's welcome.
Thank you. And let me share my screen first. Okay, guys, can you see my screen now?
Yes.
Yes.
Okay.
Yes.
Great. First, I would like to thank Dr. Jiaming Chen for the kind introduction, and also I would like to thank the organizing committee for organizing such a great opportunity, platform or series for the young PI like me to communicate and share my studies to the researchers all over the world. So today my topic is about the adaptation of Cryptococcus neoformans to genotoxic stresses and hypoxia. So now I am just establishing my own lab in the Henan Agricultural University. So this is our new building. It looks great. So all you guys are welcome to visit us. Oh. Let me see. Okay, now. So I work on Cryptococcus neoformans. So this is a human fungal pathogen with
对，刚徐总放了一个
Hello. Okay.
It's okay. You go ahead.
Okay. So this fungus has fascinating morphogenesis. So here I'm showing you the different morphotypes of Cryptococcus neoformans. So this fungus can grow as yeast when the nutrition is rich. So and also it can generate the hyphae or pseudo hyphae. So when the nutrition is get limited, this fungus can develop the fruiting structure at the tip of or in the middle of the arrow hyphae, and then to produce the infectious spores. So here through the SEM, we can see the yeast form of Cryptococcus neoformans is covered by the fibers, which are the capsule, the polysaccharide capsule. Here you can see the hyphae form of Cryptococcus neoformans. This is the feature of a crypto hyphae, which is the clam cells. So this is on the tip, at the tip of the hyphae, this is about to develop the fruiting body, which is the basidial head. Now here you can see on the basidial head, so during the morphogenesis, the 4 chains of spores can be produced. As an environmental fungus, it is ubiquitous in the environment, and most commonly it is in association with the pigeon guano, and also the different tree species. So upon inhalation, desiccated yeast cells or spores can get into the host, the human, and deposit in the lung. So in the healthy human individuals, the crypto cells can stay in the lung for decades without causing any symptoms. Once the immune system of the host get compromised, so it can disseminate through the bloodstream and get into the central, can pass the blood-brain barrier, cause the fetal meningitis. So the interesting question I'm interested in is so how this fungus become establish the latency during the infection in the lung. So there's accumulating evidence suggests that the Titan cell may be the potential dominant form of Cryptococcus neoformans during infection in the lung. So Titan cell is another, you can see it is another form or morphotype of crypto cells. So normal crypto cell yeast cells are haploid, which is smaller, and the Titan cell is larger in size and also with polyploid DNA content. And those Titan cells are resistant to various stresses, and also it's because it's big, and also it can prevent the phagocytosis from the macrophage cells. So that makes it pretty resistant to the host stresses, and also can survive in the lung. And also another, more importantly, this polyploid Titan cells was found to be able to generate the normal haploid cells during in vitro experiments. So that suggests this Titan cells has the potential to get resistant to host cells, and also can get reactivated to cause the systematic infection. So the two questions that I'm interested in is, so what kind of stress or signal triggers the Titan cell formation? So since Titan cell can generate normal haploid cells, so how is the ploidy reduction achieved? So the first part of my talk is about to answer answering these two questions. So we know all kinds of various stresses or conditions can cause the Titan cell formation, such as the nitrosative stresses or oxidative stresses. But eventually, also based on the research from like Candida species, eventually it is the genotoxicity caused by these stresses that may trigger the ploidy increase and also the cell size enlargement. So then the first experiment that we did is we tested the different genotoxic stresses on the effect on the cell size. So here we see the UV and MMS usually cause the site mutation on the genome, and also gamma irradiation and deletion, which is a drug that can cause the double-strand DNA break. So here, based on this data, you can see the gamma irradiation and the deletion cause significant cell size enlargement. So that suggests that the genotoxic stresses that cause double-strand breaks triggers cell enlargement in Cryptococcus neoformans. So takes advantage of the deletion model. So we checked the cell size and also the ploidy and the microscope. So we use GFP fluorescent tag to label the histone H2B components to represent the DNA content indirectly. So here you can see after the deletion treatment, the normal yeast cells become larger, and also the DNA content based on the fluorescence is increased. So we analyzed the untreated and treated cells through the cell analyzer. From the x-axis, we can see the cell size is enlarged after the deletion treatment. And also from the GLP fluorescent intensity, we can see the treated cells has stronger fluorescence, which means it has higher ploidy of DNA content. So that means the deletion treatment triggers Titan cell formation. So another interesting experiment we have done is like we asked whether what will happen if we transfer the Nielsen treated cells to the drug-free media. And then we found that, so see, after the treatment, you see the cells become larger, and the DNA content became most of the population of the cells have higher DNA content. But after release to the drug-free condition for 24 hours, so the cell size went back to the normal, like as normal as the untreated control cells, and also the DNA content also reduced to the similar level as the wild type control strain. So that means the Nielsen-induced titan cell formation is reversible. So this is give us a very good platform to study the titan cell formation and the reactivation process. So then next we want to know how the ploidy reduction happened. We know Cryptococcus neoformans can undergo sexual reproduction. Here I take unisexual reproduction as an example to show that so the 1C haploid yeast cells can undergo endoreplication and also pre-meiotic replication to generate 4C DNA content nuclei in the Bacillia head. And one round of meiosis can generate haploid, can mediate the ploidy reduction from 4C to 1C and generate spores. So that means in this process, meiosis mediates the ploidy reduction. So we hypothesize that whether meiosis also contribute to the ploidy reduction in titan cell. Then we want to block meiosis by deleting the meiosis-specific genes, which is DMC1. DMC1 is the meiosis-specific recombinase. When we delete DMC1, and then we can see the crypto still can develop the Bacillia head, but cannot go through this meiosis and to generate the haploid spore chains. So that means DMC1 is specific to the meiosis. And then we did the same experiment with DMC1 deletion mutant. So here we can see after the Nielsen treatment, the DMC1 deletion cells still enlarged in size, and also the DNA content increased. But after 24 hours of releasing to the drug-free condition, most of the population of the cells still shows larger size than the control condition, and also the DNA content also shows still at the 2C level, so which is different from the wild type strain that we see previously. So that means the blocking meiosis delays the size and the ploidy reduction in Cryptococcus neoformans after the Nielsen treatment. So then we want to know what happens if we block meiosis to the cells during cryptococcus infection. So our collaborator Chao Yang from Rutgers University, he infected mice with wild type strain and also the DMC1 deletion strain, and he collected the cells from the ball fluid and quantified the cell size of the population. Here, based on the staining, you can see in the DMC1 deletion infected mice, he collected the tissue and observed more proportion of the cells with larger size, and also through the quantification we can see blocking meiosis increases the proportion of large cells during infection. So that means even in the animal, in mice, so the meiosis or the DMC1 play roles in regulating the titan cell formation. So we want to detect whether the meiosis or meiosis-specific gene are expressed or activated during infection. Since the meiosis is a transient process and also the heterogeneity during infection, so it's kind of difficult to catch the meiosis event. So to achieve this, we designed a flip reporter system. And then we use the meiosis specific promoter to drive the expression of the recombinase, which can recognize those loci. And then if the meiosis specific promoter promoters get activated, then the recombinase can be produced, and then it can flip this drug marker. And then transient activation can cause a permanent like phenotype that we can screen. So with this system, first we tested this reporter system in vitro and under the like meiosis suppressing conditions and also the meiosis inducing conditions. Here we can see under the suppressing condition, there's no flip event. We didn't detect any flip event. Under the meiosis inducing conditions, we detect the plenty of the meiotic event. So that means the system is responsive to the meiosis and it's very specific. So then we use this reporter string. We construct the reporter string and then we infected mice and then we did isolation of the drug-resistant progeny from the tissue, the animal tissues. Here we use the DMC1 promoter to construct this reporter string, and in all 4 mice, we can detect the flip event. So that means the DMC1 gene was activated during infection, and also we use another meiosis specific gene promoter, REC8, also did the same thing. We got the same results. So this data suggests that the meiosis specific genes are activated during cryptococcal infection. So from this part, the take-home messages are the genotoxic stresses that cause double-strand DNA breaks, triggers tetanization. And also the meiotic machinery is involved in ploidy reduction of tetan cells in Cryptococcus neoformans. So next, I'm going to transfer to another topic about hypoxia adaptation. So as I showed you previously, so during this process, as an like environment fungus, so Cryptococcus neoformans in the environment, it prefers to have the atmosphere oxygen level to achieve the optimal growth. However, when it gets into the host, and then the oxygen levels change. So at the initial deposition site of the Cryptococcus neoformans in the healthy human lung, the oxygen level is about 14%, which is lower than the atmospheric oxygen level. And after dissemination in other like normal mammalian tissues, like in the kidney, some other tissues, so the oxygen levels will drop down to like 2.5% to 9%, which is significantly lower than the favorite oxygen level of Cryptococcus neoformans. So during along with the infection, so the inflammation or the necrosis caused by the infection, it can cause like an increased degree of hypoxia. Then the oxygen level can be in that necrosis, the oxygen level can be as low as even lower than 1%. And then, but crypto can survive under these low oxygen levels. So how this adaptation is established is the question I would like to answer. So first, to address this question, so we did a transcriptome analysis. We cultured the wild type cells under hypoxia and normoxia. We compared their transcriptome. Here you can see lots of genes get upregulated with very high fold change. And when we take a further look at like a close look at of these genes, we can see those genes are involved in different metabolic pathways, especially for the glycolysis and fermentation, and also like the TC cycles. So they suggest that suggested that under hypoxia, Cryptococcus neoformans changed or reprogrammed its like metabolism, try to adapt to the hypoxia condition. So this metabolic reprogramming adaptation to hypoxia is similar as what people observed in the mammalian cells. So in the mammalian cells and hypoxia, also the glycolysis and the fermentation pathway is activated. So if in mammals, so those processes are regulated by two types of transcription factors, which is the hypoxia inducible factors, alpha and beta. So and the normoxia, the HIF1 alpha is like hydroxylated by the oxygen dependent hydroxylase. So and then it will undergo degradation pathway. But and the normal and the hypoxia, then the PhD activity is gone. Then the HIF1 alpha can be stabilized and translocated into nucleus and form the heterodimer with the HIF beta component to upregulate or to turn on the gene regulation of this metabolic reprogramming. But these hypoxia-inducible factors are missing in lower eukaryotes. It hasn't been, there's no like homologs can be found in any yeast, any lower eukaryotes like yeast or even or also Cryptococcus neoformans. And then based on the domain, the pest domain in the hypoxia-inducible factors, we did a systematic analysis of the pest domain containing proteins in Cryptococcus neoformans, try to see whether there are pest proteins may be involved in hypoxia adaptation. Fortunately, we find one like a protein we named the PAS2, which has the pest domain and also a zinc finger binding domain. And then we tested its function in regulating hypoxia growth. Here we can see when we knock out this gene, and the hypoxia growth of Cryptococcus neoformans is reduced, and it can the white type the PAS2 regulates hypoxic growth in Cryptococcus neoformans. And then this is the picture I showed you before. Here are the metabolic genes that are upregulated when white type is cultured under hypoxia. But when we knock out the PAS2, and then we compared the hypoxia, the transcriptome with white type and hypoxia, we can see almost all those metabolic genes cannot be induced in the PAS2 deletion mutants. So this strongly suggests that PAS2 regulates the hypoxic induction of metabolic genes. So next, we did proteomics, try to characterize the potential like interacting partners of PAS2 and to reveal the regulatory mechanism behind PAS2 on the metabolic reprogramming. Here the green part are the normoxia specific potential interacting partners. Those are the hypoxia specific potential interacting partners. We are particularly interested in the three purple ones. So those are the transcription factors. So we the hypothesis is those transcription factors most likely interact with the PAS2 together in regulating the hypoxia adaptation. And then we test the hypoxia growth of those transcription factor deletion mutants and compared to the PAS2 deletion mutants. Here we can see the RDS2 deletion mutants shows very similar phenotype as the PAS2. And then we labeled RDS2 with green fluorescence and PAS2 with cherry fluorescence. Those two co-localized in the nuclei. And also we use the immune green the trap to do the pull down assay and tested the interaction between PAS2 and RDS2. Here we can see they interact with each other. And in the other we did pull down with PAS2, and also we detect the RDS2. So that means this PAS2 interacts with RDS2 together in regulating hypoxia growth in Cryptococcus neoformans. So based on those preliminary data, we proposed a model like under normoxia condition, the metabolism is normal as normal as like this, and also the ATP is produced through the the oxidative phosphorylation. But when the oxygen level gets reduced or under hypoxia, the oxidative phosphorylation process will get like suppressed to prevent the accumulation of ROS. Then that means the previous like metabolic pathways need to be reprogrammed. So PAS2 and RDS2 together, they mediated the reprogramming of these metabolic pathways, try to reduce the supply of NADH from TCA cycle to the oxidative phosphorylation, and then change the ATP, the way of producing VTP to glycolysis and also the fermentation pathway. And those the glycosylated cycles also are changed to balance the NADH and NAD and NADH level in the cell. So based on those, we are going to the future research will be focused on the metabolites during this reprogramming process. So I would like to thank my postdoc supervisor, Xiaorong Lin, and also the lab members in Lin Lab. So I developed my career there and get interested and get trained in working with Cryptococcus neoformans. Also, I'd like to thank the collaborators, Dr. Chaoyang Xue at Rutgers University, Lin Qiwang at the Institute of Microbiology, En Ce Yang at Peking University, and also Xinping Xue at Nanchang University. Those are my current lab members. I also would like to thank them. So thank you for your attention. With that, I would like to take questions later.
Right, thank you so much.
Thanks. Yeah, so Rachel, you go ahead.
All right, thank you, Changbin. So hi, everyone. My name is Rachel, and I'm from the University of Cape Town. And my task is to introduce our next speaker, who is Benson Musinguzi. Benson is a lecturer of mycology at Kampala International University in Uganda, and also Muni University in Arua, Uganda. He has worked as a laboratory services advisor in infectious diseases institute at Makerere University. And he has supported the Ugandan National Health Laboratory Services Department quite a bit. And he's a holder of a bachelor's degree in medical laboratory sciences and a master's degree in medical microbiology. And he's currently studying for his PhD in biomedical sciences at Makerere University, also in Uganda. His special interest is molecular mycology. And Benson says that he's also registered and licensed as a microbiologist with the Uganda Allied Health Professionals Council. Benson, if you can share your screen. Benson? Can you hear me? You're on mute. Great, thank you. So the title of his talk is Antifungal Susceptibility Profile of Candida albicans isolated from HIV patients with oral candidiasis attending antiretroviral therapy clinic at a teaching hospital in southwestern Uganda. If you're ready, Benson, you can go ahead and present. Thank you.
Thank you very much for the opportunity. This is Benson Sings, a PhD fellow at Makerere University. I'm glad to be part of this seminar. As my title reads, Antifungal susceptibility profile of Candida albicans isolated from HIV patients with oral candidiasis. First of all, the part of the background is that this oral candidiasis is the most prevalent opportunistic fungal disease in HIV patients, more especially in developing countries like Uganda.
Sorry,
and
Benson?
approximate, yes, please.
We can see your screen, but would you mind just putting it in presenter mode so that it just fills the whole screen up? Sorry to...
Okay. I
Awesome.
think there is
Thank you.
okay enough.
Right.
Okay, yeah, okay. Thank you. I was saying this oral candidiasis is a very common prevalent fungal infection among this cohort of patients, the HIV/AIDS patients, and it occurs in more than 95% of HIV patients. So it's associated with the morbidities, and severe cases may go into invasive disease. As in Uganda, as per the last year report, over 45,000 HIV patients have got OC-related issues, and the study that was done in all TASO centers, which look after these HIV/AIDS patients, prevalence of 14% has been recorded in this TASO center. Then with the Mbarara region recording the highest with 43% as compared to as compared to Hoima Regional Referral Hospital with 24.9%. So these several species of Candida, they cause this sort of oral candidiasis, much as the leading cause usually is Candida albicans, and these other non-Candida albicans have been recorded. These Candida albicans, there have been a report of resistance in fluconazole and itoconazole antifungals, much as these are the drugs that are commonly used in the country. And among this cohort of patients, fluconazole is usually used for prophylaxis. So now we are seeing non-albicans Candida coming on board. However, the lab diagnosis is not usually done to rule out these sick albicans and non-Candida albicans. So much as this lab diagnosis, little that is done, you find it's conventional. They use conventional phenotypic methods, which are less sensitive to detect non-Candida albicans. Still, when you are to look at the guideline, the national guidelines, clinical guidelines, it's not clear on the management of oral candidiasis. So empirical therapy at the end of the day is the most preferred without the proper diagnosis, much as these Candida species, they have differing antifungal susceptibility profile for these antifungals. So this still empirical treatment without the lab diagnosis, at the end of the day, it drives the antifungal resistance. So this study really wanted to look at how these antifungals are susceptible to Candida albicans from this cohort, from the HIV patients with oral candidiasis attending this art clinic at Kampala International University Teaching Hospital, which is located in Western Uganda. So the design, this was a one-time district correct sectional study, which was conducted among 280 patients who were attending this facility. So it was a purposeful sampling, and we targeted those who were 18 years and above, and those who consented to participate in the study, where questionnaires were used, structured questionnaires were used, and samples collected and processed to identify Candida albicans. So these oral swabs were taken using a sterile cotton swab. Some specimens were transported from the art clinic to the microbiology lab at 2 to 8 degrees centigrade and within 24 hours. And these swabs were cultured on subrhodextrose agar, being the specific for fungi, and chromo agar Candida was used of course, to speciate the Candida albicans. So this phenotypic culture and biochemical tests were also done to make sure that the Candida albicans are well speciated before carrying out antifungal susceptibility test and which was done using diffusion, disk diffusion methods. So as they flow, these HIV patients, of course, were all of them who were attending this clinic. Oral swabs were taken. After oral swabs being taken, they were taken to the microbiology lab for speciation of these Candida species. Then after that, the antifungal susceptibility profile was done to look at the antifungals which are susceptible, then also to rule out these resistant species, to rule out the resistant Candida albicans. So after the laboratory analysis, it was realized that 33 out of 280 samples, 33 samples, these are of course 33 patients, were had Candida albicans and non-Candida albicans. Then still, as part of the 33 patients, that is 33 samples, 29 of them had Candida albicans, and 4 had non-albicans Candida. So meaning that of the 11% of these people who had oral candidiasis, Candida albicans contribute.
Benson, you're muted.
Yes, Candida albicans contributed 88% among these OC patients, while the NAC, that's non-albicans Candida, was contributed 12%. Literally meaning now we are seeing the non-albicans, which sometimes are very hard to treat. They have got a high resistance is coming on board among these patients, yet the diagnosis is not well done. Then the distribution of Candida albicans among patients attending at clinic, as I had said before, when you are to look at the individual distribution, is that these Candida albicans among the 280 patients who are enrolled in this study, it contributed 10.4%, because there were 29 samples. Then the non-albicans Candida contributed 1.4%. So looking at how all these, Candida albicans responded to the commonly used antifungals that we have in the country, still most of the antifungals were susceptible, meaning they were really working well on these Candida albicans, except the fluconazole, which was the susceptibility was at 10.3%. Literally meaning that this fluconazole is is highly resistant, actually 90% resistant, plus fluconazole. Much as this fluconazole is the drug that is commonly used on this cohort of patients, and at the end of the day, that's what is used for prophylaxis, especially in meningitis, patients suspected of meningitis. They usually use this fluconazole. So we ask ourselves, why could this be the results of the results? Why did they look like this? And the 11.8% prevalence of this oral candidiasis is slightly low compared to these previous studies that were done, but most of these studies were on clinical aspect. They were not looking at the laboratory diagnosis. They were looking at the clinical diagnosis, and we think that it could be the reason. It could be the reason why the prevalence was slightly lower than what was reported previously in Western Uganda. However, there are other reports that have reported similar results. And at the same time, this study excluded children. So much as it has been reported that the children have got high colonization of Candida albicans. Then still, we concentrated on patients who were taking this antiretroviral therapy treatment, literally meaning that the candidiasis is already suppressed at some point. Then this high resistance of fluconazole up to 89.7% is also in agreement with some studies which have reported higher resistance in fluconazole. And this may be due to over-dependence on fluconazole for treatment of oral candidiasis in Uganda. Yeah, thank you. Thank you very much. That's what I had for this morning. Thank you very much. Responding to questions.
Thank you so much, Benson. And I think now it's time we go to the questions and discussions around the two presentations. Changbin, would you like to start?
It's okay. Okay, thanks, Rachel. So I think there's in a checkboard, there's several questions listed for Youbao. So I think the first question is what's the virulence phenotype of DMC1 mutant?
We did test the virulence effect of DMC1 deletion. We didn't see any difference in terms of fungal burden and also the fungal load in the brain tissue. And we didn't test the survival, but we think there should be no difference with Y type compared to Y type.
Okay, I think the second question is based on your neomycin treatment. You say that when the cells released from the neomycin treatment,
yes,
and looks like the 1C population increased and different from the initial state. So how to explain this phenomenon?
Okay, I think based on the data, you can see the height of the peaks are different, but that doesn't mean the populations are different. So that means that for the control cells, let's say the initial state, the cells were cultured for 12 hours. So the cells, most likely the cells were still at the active, like propagating state. And also that's why you will see the 1C, 2C dynamic. But for the releasing experiment, the cells were released for 24 hours. So that means maybe the cell at that time was not that active in reproducing or embodying process. So that makes you feel like the peaks in the effects in the cell analyzer shows difference, but that doesn't mean the 1C population is increased. That's just the population difference within the treated or the untreated. So it's not comparable maybe from the initial state and also the release cells, since they are at the different culture status. Yeah.
Right. Changbin, I'm just going to ask here a question for Benson. Okay. Benson, there's a question that says, what are some of the NAC species isolated in this study?
Thank you very much. Right now, that is actually what we are working on, because this study was a baseline for looking at now a speciation of these non-Candida albicans. So far, we have isolated Candida cruzi to pick Candida tuberculosis and dubriscens. Then we after that, we have not yet done the antifungal circulated profile to look at it. But now this study is ongoing, and because we are looking now at the molecular component of it, to look at the resistance patterns and probably we shall also go ahead to look at the mechanism of this resistance. So far we have isolated Kruse, Tupkas, and Benson. Thank you.
All right, thank you. Changbin?
Yes, yeah, and the next question is, I think is, what's that say? You said why the DMC mutant, the induction rate is low in V8 condition.
Okay, the number is small, because I think this is the experimental limitations. Since we know if you work on the crypto, we drop the colony on the V8 and then we wait for a week or a few days to wait for the sporulation, and then we collect the edge as good as we can to collect the spores from the edge, but we still cannot get rid of most of the yeast. So that's why it makes most of the cells still are the yeast cells that we collected from the wheat plate. So that makes the numbers, the total numbers big, very big, but actually we didn't collect many spores. So that's why it looks like the flipped or the resistant progenies on wheat conditions is lower compared to the total CFU. But I think it's because of the experimental limitations. We cannot get clean like hyphae and spores without yeast.
Okay, so Rachel, do you have a question for Benson?
There's a question here, Benson, and it says, did you see any differences between drug sensitivities of the patients on ART versus those not on ART at oral candidiasis diagnosis?
Thank you very much. We actually had no control group of those people who are not on ART, but when we considered the results from the registers respectively, I would say that there is a very big difference, especially in fluconazole resistance, and that could be explained by the fact that these people on ART, they are taking this drug for prophylaxis. So there was a significant difference, much as this was, it wasn't analyzed in our study. It wasn't analyzed to look at the significance of the results. Yeah, thank you.
Yes, okay, I think one question from Jessica. So she asked, what's the morphological and the metabolic difference about the mitochondria of Titan cells look like compared to the normal cells?
Thank you. It's a very interesting questions. So the quick answer is we don't know. We haven't checked this, but I think it's worth to check the mitochondrial morphology. It should have changed since the mitochondrial morphology is tightly associated with the stress response and also the Titan. Maybe in the Titan cells, it has like specific features in terms of mitochondrial morphology. And also the metabolism should also be different in Titan cells, since Titan cells are not actively like in reproducing. So there should be metabolic. I mean, the metabolism should be different in Titan cells. It's a very interesting question. We definitely would like to check. Yeah, thank you.
Yes.
Right. I don't have a question here for Benson, but I just want to ask you, Benson, as a follow-up from the last question on ART, how compliant are the patients that you, like your cohort, like what percentage of them were compliant to the ART treatment? And does that have an effect on your observations, do you think?
Thank you very much. These patients, of course, they are compliant. They are taking the drugs well. And of course, as part of this study, it went through previous approvals, ethical approvals. And of course, participation was voluntary. But most of them, actually I may say 90%, accepted to participate in the study. So I don't think it had a negative impact on the results of this study. Thank you.
Okay, I think one more question from Linqi. So he asked the let's see. It looks like the DMC not mutant only reduce the 1C progeny, but not a abolishment. So does that mean there's a meiosis activity independent mechanism about this ploidy reduction of Tatten cells?
Yes, it's a very good question. So our idea is like a meiosis mediated ploidy reduction, like during infection or in the Tatten cells, it's only very small, only contributes a very small portion to this process. So it also shows in our data from the animal, from the flip reporter system, we detect a very small amount of like flip or resistant event. So that means only a very few proportion of the cells undergo ploidy reduction through meiosis. So most of the cells, or most of the Tatten cells, they undergo ploidy reduction maybe through the parasexual cycle, like a random chromosome loss. So that is still, let me simulate in Candida, that is still the main route for the ploidy reduction in Tatten cells in crypto also. So meiosis only contributes part to this process. Definitely there is meiosis independent pathways to mediate the ploidy reduction in Tatten cells. Thank you.
Okay. All right. Thank you, Youbao. A question for Benson, and it says, when an oral candidiasis infection goes systemic, is it known how it tends to do so? As in, does it go through the cribriform plate or through the bloodstream?
I beg your pardon.
Okay, the question says when there's an oral candidiasis infection and then it goes systemic, that means it spreads now. How does it tend to do so? Does it go through the cribriform plate or bloodstream?
Thank you very much for this wonderful question about the pathogenesis. This study actually has not really extended deeply to look at the pathogenesis about how the spreading is done. That would be actually a wonderful research question for the next study. So during actual publication of this work, these are some of the things we could recommend for the further studies.
All right,
thank you very much.
Changbin?
Okay, I have still a question for you, Bob. So is the question is whether whether the DNA recombination occurs during DMC not mediated ploidy decrease, and can we view this process as a form of sexual reproduction?
Thank you. So yeah, this is an interesting question. So we also thought about this, and then we also designed the experiment to try to detect whether DNA recombination occurred or not. And the idea is like we made a diploid cell with flip reporter, and we infect mice, and then we isolated the resistant flip progeny, and then we did genome sequencing, try to see whether there's DNA recombination happens. So now we are in the process to analyze this genome sequence data. But at the preliminary results, we can see there are DNA recognitions occurs across the chromosomes, especially in some specific locus in the chromosome 11, I think. But we also tried to validate this sequencing data by like RFLP, like vet experiments. But we haven't successfully like very validate any recombination loci. That's why we maybe need this might be due to the sequencing, since we use the Illumina platform did the sequencing. So for detecting the recombination, we may need the long reads like the PacBio or nanopore sequencing to see whether there is the recombination occurred or there is like a conversion events. So that's definitely in the plan. Yeah. Thank you.
Thank you, Youbao. The last question here for Benson says, can you just comment on the mean inhibitory concentration of the drug you used for resistant strains? I've tried to paraphrase the question as best as I could. Just comment on the mean inhibitory concentrations of the drug concentration you used.
Thank you very much. We are, of course, we are using disc diffusion for these different drugs, much as, of course, it's not a standard method in our current study. Actually, we are evaluating this method, the disc diffusion method, using the, actually, we are comparing it with the e-test to see which one works well. Then using broth diffusion method, broth concentration method to this. They have got different concentrations, but the most recommended by clinical laboratory institute standards is the broth concentration method. Thank you.
All right, thank you.
So I think no more question right now. So I think we just wanted to thank the two speakers and all the audience today. So I think, Jessica, do you have any announcement?
No other announcements other than please submit abstracts if you're interested. We're looking for speakers both at the other time and at this time. So whichever one is more convenient for you, just submit an abstract and let us know. We had a great response to this, so we'd love to have more speakers who are interested in this time. And then I've also put the information about the mycotox in the chat, and the next one is 2 weeks from today. So hopefully we will see you there. But thanks everyone for great talks, and thanks for everyone for coming.
Thank you.
Thank you very much.
All right, thank you.
Bye-bye.
Thank you. Yeah, thank you very much.
Bye.